Yeah.
So going back to this, guys, so they actually collect the bacteria in whatever specimen, and then they incubate it at this particular temperature. So always have that at the forefront of your mind. So collection is like the first thing, all right? Now, in terms of the shapes of the bacteria that we're dealing with, these are the most common ones, all right? So you're gonna have your coccus, which is a spherical shape. You have your rod-shaped bacillus. You have your spirillum rather. You have your diplococcus, which is basically, of course, two cocci. You have diplobacilli. You have your staphylococcus, which is clusters of coccus. You have your streptococcus, which is basically, a chain of cocci. And you have your streptobacilli. The ones that you guys must pay keen attention to, especially these two, the bacilli and the cocci. To a lesser extent, the spiruli, but these two are really common in terms of the organisms with which we have to deal with, not only for examination purposes, of course, but also within the medical field, within common exposures and things which really and truly cause infections. And of course, this is basically the arrangement of the cocci. So you have your coccus, you have your diplococci, which are two. You have your staphylococci, streptococci, which I mentioned before, we're assuming the orientation of chains. And to a lesser extent, we can, not really and truly pay much attention to the sarcinae or the tetrads. So you have fewer organisms, assuming that shape that we have to deal with. So in terms of the classification of bacteria, there are two in terms of that. So you're always going to have to deal with either your gram-positive or your gram-negative. And these names are actually given to the bacteria based on their response to staining. So a gram stain test will be conducted, and based on their properties, based on the properties exhibited by the bacteria in response to that test, you're going to now have a result that would either be positive or negative. So how do we identify the types? So as I mentioned before, a gram stain test is actually done. Now is that which basically give a positive result to the gram stain. And as I mentioned, it is actually a principal crystal violet dye that is actually applied to it. And then this is the peptidoglycan wall. So it is very thick. And there is a picture in the subsequent slide which will actually show a comparison between the gram positive cell wall and the gram negative cell wall for you guys to have a clearer picture of what we are dealing with. So when seen under microscope, it appears to be purple because of this. So it retains the violet color. It has a very thick peptidoglycan wall. So it is able to retain that stain. Where the gram-negative bacteria is concerned, though, they have a very thin peptidoglycan layer. And that means that that principal stain will be seeped through. So it cannot really and truly retain the stain when it's applied. And so once you actually decolorize it now with the alcohol-based material, for example, it actually degrades the membrane. So this is a cell membrane, it's the inner membrane. All right, guys, this is the inner cell membrane. This is the outer cell membrane. So what happens is that after the decolorization phase, as I mentioned before, you could use an alcohol-based product. It is actually going to degrade this layer. And since this wall is actually relatively thin compared to the gram-positive organisms, that which belongs to the gram-positive organism, it has no other option now but to seep through. So the principal stain that was used, much of it will not be retained. Much of that color will not be retained. And remember now, after you use the principal stain and decolorize it and everything, you're now going to apply the counter stain, and that is pink, all right? It's either red or pink, and so only that color will remain because you will not subsequently decolorize it after the counter stain. That is it for the process of the gram stain test. So as I mentioned before, the gram-positive bacteria now, they will retain the principal color, retain the crystal violet, and to a less extent, the safranin will not be brilliant in terms of the intensity of the color to override that purple color. But the gram-negative now, it is actually red or pink, all right? So this is what I was referring to. So you have that cell membrane for the gram-positive. You don't have an outer membrane for it. And you basically have that very thick peptidoglycan cell wall, as opposed to a very thin peptidoglycan cell wall in the gram-negative cell. So once this is actually decolorized by that alcohol-based substance, you're now going to have that color seeping through because it degrades the outer membrane, and therefore you will not have that color remaining. And so only the safranin, the counter stain, will be exuded from it, all right, in terms of the color that's present. So this is how it actually looks in terms of a gram-positive bacteria, all right? They're retaining that principal crystal violet color, as opposed to a gram-negative now, who, because of their thin peptidoglycan layer, they now only have that safranin color. And that is really and truly the difference, which allows us to identify whether it be a gram-positive or a gram-negative. So growth mediums, we're going to actually delve into that now. So, of course, now, guys, there are actually 3 growth mediums that. Yuck. Why are you saying that, Bianca? You know there are a lot of those things inside of you. I don't need to tell you that, ma'am. You know a lot. You know they reside within us. So yeah, guys, there are actually 3 growth mediums, all right? And the first being the blood agar, you have the chocolate agar, and you have the McConkey's agar as well. And there are other culture medias that are used, and you have one main one known as the India ink preparation. We'll delve into that later down in the presentation. But the 3 that we are going to be focusing on right now are actually these listed above: blood agar, chocolate agar, and McConkey's agar. Right? So the blood agar, you know, it's a really enriched media. And as it says, it contains 5 to 10% of blood. So it predominantly, guys, have red blood cells. And it also has like nutrients within it. And the blood is normally taken, as can be seen here, from either a horse or sheep, all right? So what is actually done here is that it actually targets bacteria that would damage red blood cells. So it therefore cause hemolysis. And I'm sure, you know, we all perhaps have an idea, even though if we didn't hear it, I'm sure we've heard the term before, but hemo, you know, means blood, and lytic would mean lysis, which would mean, you know, it's actually split or it's destroyed, basically. So hemolysis. So it's basically targeting bacteria now that would cause the blood to actually lyse, all right? So these are actually particular organisms that would, you know, be grown on these media, these type of media, because of what they would exhibit in terms of the pattern of lysis, of course. And these are both fastidious organisms. Does anyone know what a fastidious organism is? Have we ever heard the term before? Nope. Ariane, have you ever heard it before? Of course, of course she's heard it before. She did microbe. Any ideas? Of course. So Ariane knows what it is. So a fastidious organism, guys, is just one that really and truly requires particular nutrients, and so it needs nutrients and a particular environment in order to grow. So it won't just grow. Good, very good, Ariane. It needs complex nutrients, as she mentioned. So you can't just grow on any and anyone. Yeah, the really picky ones, Jada, really, really picky ones. So like the streptococci and the Neisseria, which cause gonorrhea, these are fastidious organisms. They really and truly will not grow unless you actually give them the certain nutrients so that they can exhibit that growth properly. So as I mentioned before, you actually get lysis of the blood when these organisms now start to grow there. And based on the lysis now, you will subsequently have different patterns of the hemolytic process occurring. So for beta hemolysis, B, so just remember guys, B means beta. That's how I remember it at least, and perhaps most of us. So you see that group A and group B strep, they are both beta hemolytic. So just remember B means beta, all right? And a thing that we can pick from here is that it's actually the brightest in terms of the hemolysis that ensues from it. So it's the brightest pattern that we're going to get when these organisms start to lyse the red blood cells that are contained within the growth medium. For alpha hemolysis, we can see that it is dull. It is dull with a little bright. It's a bit brighter than the gamma, but it's not, of course, obviously as bright as the beta. And the gamma, is really dull. It's the dullest of the three. And both the group D strep, exhibit both alpha or gamma hemolysis. And you're going to have that hemolytic process of beta hemolysis coming from the group A and the group B. All right. So the chocolate agar now. The thing with the chocolate agar is that it is really and truly just blood agar that is actually heated. And there's a reason for this, as we'll see in subsequent slides when we actually get to know the full reasoning behind it. So it's actually heated so that particular nutrients can be exuded from the blood so that they can be within that environment. So the organisms can now take it up. So you have some organisms which would prefer for that to be the immediate environment so that growth can occur. All right. So it's just remember, it's actually a blood agar that is heated and it gets this chocolatey appearance. And that is why it's actually deemed chocolate agar, also referred to as GC agar. And we see that particular organisms such as the Haemophilus influenza and the Neisseria species, they grow there. Okay, so just remember that. And then we have the McConkey agar. Okay, this now is actually to isolate the gram-negative bacteria. So we'll find that gram-positive bacteria cannot grow at all on McConkey's agar because it's actually designed so as to only grow the gram-negative bacteria. So if you can think about most of your gram-positive, the staph, your strep, they will not be able to grow on the McConkey's agar. Things like your E. coli and your Haemophilus influenza, those which are gram-negative will therefore be able to grow here. And the reason why the gram-positives can't grow here is that the media is so designed to prevent or to inhibit growth because it has one, the bile salts, all right? So the bile salts, as we see, will inhibit the growth of the gram-positive organisms. And you also have Kristoff violet dye. And these two things actually prevent the growth of the gram-positive bacteria. The thing as well with the McConkey's agar is that it allows you to see if the bacteria, the gram-negative bacteria, is fermenting the lactose that it has, all right? So the media would have that lactose in it. So if it's a lactose fermenting organism, you will get a particular color. It will become pink. But if not, you will have just a sort of yellowish color that's there, and that would indicate that the organism is not fermenting the lactose. All right, let me check the chat. Well, all right, so in terms of the incubation period is basically just going to tell you really and truly how long the bacteria will grow. So it is necessary now, yeah. It's not going to tell you no. So just think of incubation in terms of the period of time that it's going to take for that bacteria now to start showing, for you to start seeing signs. All right, that is basically what incubation is. So some organisms take perhaps 12 hours on average, 12 to 20 hours to grow. We have some other organisms that are a bit, how should I put it? Stubborn. Yeah, I think stubborn is the right word. It can go up to 72 hours for you to start seeing these signs on the media. So the incubation is just the period of time that will have to elapse for you to start really and truly seeing the growth of the bacteria. The medium now is what will facilitate the growth. So is that clearer for you? Yeah, okay, perfect. So as we see for the McConkey's agar, remember we said it's just 4 gram negative because it contains the bile salts and the crystal violet dye, which will prevent the growth of the gram positive organisms. And it will also allow us to identify whether the gram negative is actually lactose fermenting, as we can see here. They assume that pink color. So your Escherichia coli, your E. coli, and your Enterobacter aerogenes, those are lactose fermenting gram negative organisms. What we can see here, the Salmonella and the Proteus, these are gram negative non-fermenting. Question, guys, why is this color different from these? Why is the color for the Staph aureus different from these? Any ideas?
It
So,
produces
all right, let me go back. So I didn't, let me see somebody.
It produces staphyloxetin, which is a golden colored pigment.
Gold? Jada, that was actually funny. No. That's not the answer. Aureus means gold. No. All right. So I haven't introduced you guys to the different organisms as yet in terms of their classification. But so let me go back a bit. I think this may give you guys an idea. So this is the initial color of the McConkey agar, right? Has that goldish tint, not really goldish, but that kind of brownish color, right? If you realize this is still brown. This is gold, gram negative, and this is pink, meaning gram negative, but it's lactose fermenting. So if you realize this is kind of just the same color as the initial material, so this tells us that this bacteria cannot grow here, right? So it tells us that the Staph aureus would be a gram positive. Because remember I told you guys that the McConkey agar is so designed as to prevent any form of growth of a gram positive organism. So this color, this gold color, would mean that the gram negative is not fermenting. But we can realize that for both types of growth that occur for the gram negative, you either have a gold color or a pinkish color. A gold would mean that it's not fermenting, but a pink that it is fermenting. But it doesn't remain as the original, because so this is really actually the original color of the McConkey's agar. So you have no form of hemolysis there. You have no color change or anything because the Staph aureus is just unable to grow on it because its growth is inhibited by the bile salts and by the crystal violet dye that is actually placed within the medium. All right. Was that clear, guys? So it's actually a gram positive organism. That's the whole deal here. These are all gram negative and the Staph aureus is gram positive. Yeah. Right. I know it's kind of unfair because you guys haven't seen the classification of this different species, but I kind of wanted to, you know.
Yeah. Yeah. Yeah.